AS Level Biology 9700, Paper 2, Variant 1, May-June 2025, question number 4. So this is the third video on this paper. So question number 4 starts as cells of the immune system have cell surface receptors that detect molecules made by pathogens. One of these cell surface receptors is known as TLR8, the gene. TLR8 is found on the X chromosome in humans. So 4.1 shows the production of messenger RNA formed from the gene TLR8 in the nucleus of a macrophage. So site of the attachment, you can see in picture the DNA molecule. Site of the attachment of the enzyme. So they have mentioned with an arrow. Then they have shown the primary transcription of DNA has been taking place. So primary transcript undergoes several modifications. Several modifications means the post-transcriptional modifications to become a mature messenger RNA ready for translation. In transcription, you know, students, the what happens, that is the DNA is copied by, and then the messenger RNA formation takes place. So this is the transcription. So here you can say the modificational changes are taking place into this messenger RNA. As for example, on 5' end of DNA, you can see a cap, a modified guanine nucleotide is added to the 5' end, and on the 3 polyadenylation, a long chain of the adenine nucleotides, a chain of the adenine nucleotide has been added. This is the 5' end of DNA, and this is the 3' end of primary transcript. On 5' end side, you can see a cap has been added. On the other hand, this cap is guanine nucleotide. And on the other hand, adenine tail has been added. So then what happened at stage Y? And stage Y, you can see non-coding sections, which are called as introns, are represented over here on this with the darker blocks. So they will be removed, and the coding sections exon, which are coded, represented by light blocks, are joined together. So in this way, this whole process has been taking place in figure 4.1. Now the question is, name the enzyme that catalyzes the transcription of DNA. This is very straightforward question. After this long description, that is the name of the that enzyme which will catalyze the transcription of DNA, and the name of that enzyme for this purpose, for synthesizing an RNA molecule from a DNA template, is RNA polymerase. So you know the messenger RNA will be formed over here. So for this RNA polymerase. Very simple question. Now, moving towards the question, part 2 of this question. After the name of the enzyme, which is used for the transcription. Now, the figure 4.1 shows the primary transcript is modified by the addition of nucleotide to both ends of the molecule. The cap shown in figure 4.1 is a guanine nucleotide that is added to the 5' end of the RNA, and the poly tail added to the 3' end, and consist of many adenine nucleotides. You had seen many. So the cap and the tail have a function in stage Y and are also important for the stability and the role of messenger RNA. And you know the stage Y is splicing. So during the splicing, you know that exon has been they have been removed. Introns has been removed and exon has been added. So suggests the functions of the cap and the poly A tail in the stability and role of the messenger RNA. So I would like to tell you that both the cap and the poly adenine tail are crucial for the messenger RNA to be successfully translated into a protein. So structure, you can say, it protects the messenger RNA from being broken down by endonuclease enzyme that degrade nucleic acid. So it acts as a recognition signal for the ribosome to bind to the messenger RNA, initiating translation. So the tail contributes to the half-life of the messenger RNA. Longer tail means the messenger RNA persists for longer before degradation. So facilitates the export of the mature messenger RNA from the nucleus to the cytoplasm. So the combined functions ensure the messenger RNA remains intact long enough to be translated into the TLR8 protein. So I would like to write all this which I have told you, that the first of all, what will be the structure is and function is very much related. So first point which we can write, that is uh, the cap which is on 5' cap protects the messenger RNA from being broken down by endonuclease enzymes acts as a recognition signal for the ribosome to bind the messenger RNA, initiating translation for protein synthesis. And the second point, that is the role of polyadenine tail. So the polyadenine tail contributes to the longevity of the messenger RNA. A longer tail means the messenger RNA persists for longer before degradation. Facilitates the export of the mature messenger RNA from the nucleus to the cytoplasm. So in this way, both of these modifications play a very crucial role in translation, or simply, I would like to say, the process of protein synthesis. Next question, describe the process that occurs at stage 5. Already told you the process of splicing takes place at stage 5 in figure 4.1. So how do it takes place in eukaryotic pre-messenger RNA processing? It converts the primary transcript into mature messenger RNA molecule ready for translation. So before this, the messenger RNA is ready for translations for this purpose. The different steps which takes place in, first of all, is the identification of intron and exons. That is, the primary transcript is composed of exons regions shown as light blocks in the figure, and the introns non-coding intervening regions. So they show as dark blocks. So the assembling of the splicing sums takes place. So splicing is carried out by a large complex molecule machine called the splice sum, which is made up of small nuclear ribonucleoproteins and numerous other protein factors. So here, the steps removal of the introns takes place, and then the excision and ligation takes place. So I would like to write all these main points of these stepping. So splicing process takes place as under in following steps. That is, the first step is identification of introns and exons. So the coding and node coding sequence. So in this case, the non-coding sequence is removed and the coding sequence, they are added. So the second step can be the splicing is carried out in which you can see this large splicing process takes place. And then after this removal of removal of introns, which are short nucleotide sequence, which are specific short nucleotide sequence. So these are the non-coding sequence. Then after the removal, The intron sequence are and removed and then using in the same. After their removal, the exons, the fourth step is the exons are then ligated together to form a continuous coding sequence. So in this way, the process of splicing takes place. The next question is the nucleotide sequence thymine, adenine, guanine is repeated in the direction 5 to 3 in the telomers of human chromosomes. State where in a chromosome the telomers are found. So I would like to go with this question. So first of all, uh, we have to tell where the telomers are found at the ends or tips of the linear chromosomes. As you know, the telomers are present. So I would like to write over here, telomers are present at the ends or tips of chromosomes. So now the question is outline the role of telomers. So outline the role of telomere is the next question, and we are going with this. First of all, I would like to discuss the outlines. That is the first main function is protecting from degradation and fusion. So they prevent the chromosome ends from being recognized as double-strand DNA breaks, thus stopping them from fusing with other chromosomes or being degraded by DNA repair enzymes. Then solving the end replication problem as act as a buffer against the natural shortening of the chromosomes that occur during DNA replication. So by shortening the repetitive non-coding text sequence instead of essential genes, they ensure that the genetic information remains intact. So I first is they protect, they prevent the chromosomes ends from being recognized as double strand DNA breaks. stopping them from fusing with other chromosomes. Or being degraded by repair enzymes, by DNA repair enzymes. And other can be the solved, they act as a buffer against the natural shortening of the chromosomes that occur during DNA replication. So in this way, you can see during each replication, the part of the DNA can be lost. So the telomers save the DNA sequence from wastage or lost. So in this way, the genetic information remains intact. Moving towards the next question. Question melanoma. Noma is a type of tumor that develops from pigment-producing skin cells known as melanocytes. Outline how a tumor may form from a melanocyte. So, first of all, outline how a tumor may be formed. Let me move towards this question. That is the first of all, I would like to describe that how do this process takes place. So here in this case, a tumor forms through a multi-step process involving accumulation of genetic mutations. So when a tumor is formed from a melanocyte, so different steps takes place during the formation of this tumor. So first step which can be is a process of mutation. So a mutation in a proto-oncogenes. And what are oncogenes? The oncogene that promotes normal cell growth converts it into a hyperactive oncogenes. So it takes place due to the mutation, which stimulates the cell to divide to divide rapidly. So there is another type of mutation uh, which can also occur in a tumor suppressor gene. So this mutation activates the gene removing the brakes and allowing the cell to divide without regulation. So cell divide without regulation. It leads to a complete loss of cell cycle control, resulting in uncontrolled fast mitosis of the melanocyte. This uncontrolled proliferation of these abnormal cells means when there is uncontrolled division, so the cells will not be the normal cells, so it will be the abnormal cell division. So abnormal cells form a mass known as melanoma. So in this way, tumor may form from a melanocyte when there is uncontrolled cell division. Last question, part of this question, number 4. I would like to read the question first and then I will write the answer. A melanoma tumor is cancerous and may spread to other parts of the body. T-vec is a new drug that has been developed to that melanoma that has spread to other parts of the body, including lymph nodes. T-vec contains a virus that infects some of the melanoma cells, causing the cells to burst and release their contents. Some of the contents of the melanoma cells act as a cytokines and others act as antigens. Explain the effects of the cytokines and antigens released from melanoma cells in stimulating the immune system to destroy the cancerous cells. So now we have to explain the effects of the cytokines and antigens released from melanoma cells in stimulating the immune system to destroy the cancerous cells. So first of all, I would like to write the roles of what will be the roles of antigens. Antigens. First of all, I will write about antigens. The antigens the antigens which are released which are released are taken up by antigen presenting cells such as macrophages or dendritic cells. The antigen representing cells such process the antigens and display them on their surface. Presenting them to T helper cells. And then the role of cytokines. So the cytokines the released cytokines act as signaling molecules. They promote the activation and proliferation of T helper cell and cytokine T lymphocytes. And then after this, the T helper cells release their own potent cytokines, which stimulates the differentiation of cells into killer cells capable of targeting the tumor. And then which stimulates the differentiation of cells into killer cells capable to target the tumor. So the T cells binds and destroy the normal cells by releasing toxic substance. So in this way, the destruction of the cancer cells by the immune system takes place. So these are the different steps which can be written to answer this question. So it was all about question number 4. Please like, share, and subscribe my channel. So because you will be able to watch the complete paper. Thanks for watching.